Hello my dear friends, welcome. Now our topic is about hybridization. Coming to the definition, when a plant or animal is bred with a plant or an animal from a different stock, so the process which we call it as hybridization. So coming to the types of hybridization, on the basis of cell type involved in the crossing over, so hybridization is being differentiated into hybridization in sex cells and somatic hybridization. Coming to the first, so the types of hybridization on taxonomic relationship of two parents hybridization classified into two broad groups. One is intervarietal hybridization, another one is distant hybridization. First, intervarietal hybridization is again divided into intraspecific hybridization and interspecific hybridization. Distant hybridization is again divided into intrageneric hybridization and intergeneric hybridization. Coming to the first, intervarietal hybridization. The parents involved in the hybridization belong to the same species. In crop improvement program, this type of hybridization is commonly used. For example, crossing two varieties of wheat or other crops. Intraspecific hybridization. Crosses are made between two individuals of same species, such as between the Bengal tiger and Siberian tiger. These crosses are done to improve the self-pollinating crop producing also. Interspecific hybridization. Crosses are made between individuals of different varieties of a same species, such as between lion and tiger. These crosses are made to improve the self-pollination and certain cross-pollinated crops also. Mule, which is a cross between horse and donkey. Distant hybridization. The parents involved belong to different species of a same genus or of different genera. So this is classified into intrageneric and intergeneric hybridization. Coming to the first, intrageneric hybridization. Crosses are made between two individuals of different species belonging to same genera, such as between bison and cow. It is used to produce the resistance varieties for diseases, frost or drought. So beefalo, cow and crossed with bison, we call it as beefalo. Intergeneric hybridization. Crosses are made between two individuals of different genera belonging to the same family. It produces a resistance variety as well as desirable combination of all characters. For example, Brassica is crossed with Raphanus. So Raphanus brassica. Triticale, wheat plus rye is equal to triticale. Examples of hybrid organisms are a sterile trillium hybrid between Trillium and to Trillium grandiflorum. So somatic hybridization. What is somatic hybridization means? Development of hybrid plants through the fusion of somatic protoplast of two different plant species or varieties, which we call it as somatic hybridization. So somatic hybridization technique is isolation of protoplast, fusion of protoplast of desired varieties, identification and selection of somatic hybrid cells, culture of the hybrid cells, generation of hybrid plants. So isolation of protoplast, separation of the protoplast from the plant tissue. One is through mechanical method, other one is through enzymatic method. First, mechanical method. So plant tissue is being selected, so cells plasmolysis. So taking out of the plasma and microscope observation of the cell, cutting the cell wall with the knife and release of protoplasm, collection of the protoplasm. This is mechanical method through cutting the cell wall with the knife. And next, use it for vacuolated cells like onion bulb, scale, radish, etc. Low yield of the protoplast, laborious and tedious processes, low protoplast viability. Next, enzymatic method. So initially, leaf sterilization and removal of epidermis by two ways. One is plasmolyzed cells and through pectinase plus cellulase. So the protoplasm is released and the isolated protoplasm is being formed. And again through the use of pectinase enzyme, the release of isolated cells and isolated protoplasm, and again with the use of cellulase, means which will digest the cellulose present on the cell wall, and finally the protoplasm is released. So this is enzymatic method. By the use of enzymes, we are digesting the pectin and cellulase. So pectin and cellulose, and the protoplasm is being released. So used for the variety of tissues and organs, including the leaves, petioles, fruits, roots, stem, shoot, apiece, embryo, microspores, etc. The mesophyll tissue most suitable source. High yield of the protoplast, easy to perform, and more protoplast viability. So this is than mechanical advantage. The enzymatic method is highly advantageous. Protoplast fusion. Fusion of protoplast are two different genomes. So this is done by two types, spontaneous fusion and induced fusion. First is spontaneous fusion, intraspecific and intergeneric. Induced fusion, chemofusion, mechanical fusion and electrofusion. Spontaneous fusion. Protoplasts fuse spontaneously during the isolation process, mainly due to physical contact. Interspecific produce homokaryons. Intergeneric have no importance. So induced fusion. Chemofusion or fusion induced by chemicals. The types of fusogens are PEG, nano, calcium ions, polyvinyl alcohol. So we are inducing with the help of chemicals. So mechanical fusion. Physical fusion of protoplast under the microscope by using the micromanipulator and perfusion micropipette. So electrofusion is being done by fusion induced by electrical stimulation. Identification and selection of somatic hybrid cells. So hybrid identification based on differences between parental cells and hybrid cells. So hybrid selection are several markers are used. One is genetic complementation, phytotoxins, specific amino acid, auxin autotrophy, antibiotics, auxotrophic and metabolic mutants, chromosomal analysis, herbicides. Culture of the hybrid cells are cultured on a suitable medium provided the appropriate culture conditions. So regeneration of hybrid plants. Plants are induced to regenerate from hybrid cells. These hybrid plants must be at least partially fertile, in addition to having some useful properties to be of any use in the breeding schemes. Advantages of somatic hybridization. Production of novel interspecific and intergeneric hybrid. For example, pomato, hybrid of potato and tomato. Production of fertile diploids and polyploids from sexually sterile haploids, triploids and aneuploids. So transfer genes from disease-resistant abiotic stress resistance to herbicide resistance and many other quality characters are the advantages of somatic hybridization. Production of heterogeneous lines in the single species which cannot be propagated by vegetative means. Studies on the fate of plasma genes. Production of unique hybrids of nucleus and cytoplasm. Limitation of the somatic hybridization is poor regeneration of hybrid plants, non-viability of fused products, not successful in all the plants, production of unfavorable hybrids, lack of an efficient method for selection of hybrids, no confirmation of expression of particular trait in somatic hybrids. So these are all the limitations of the somatic hybridization. So these are the different types of hybridization and their methods of hybridization. So thank you very much. Please do like, share, comment and subscribe to my channel for more biology videos. Thank you.